So hi, I'm Flavia Berlinghieri, and today I'm going to talk about my project, but I also share the PhD with Paolo, so we're going to share this a lot. So we are PhD at Groningen University and Macquarie University, so we are a double PhD degree. And today I'm going to talk about one data chapter of my future PhD thesis. And it's about the stickleback parents' influence the development, personality, and brain laterality in their offspring. Okay, so I'm gonna skip the definition of laterality just to be more efficient in the time. But we can see that I'm gonna talk about visual laterality. So we hear about motor laterality before with rats, but here I'm gonna talk about visual. So which eye they use to look at something. And the main part is about the different eye that they use, but at the same time, for example, they can look at the predator. So they monitor the predator one eye, but they look for food with the other eye. So it's like multitasking capability. And then I'm going to talk about personality. It is defined as individual consistency of behavior across time, but also across context. And it can be analyzed by performing different tests to look at different behavioral axes. And one of the most studies is the axis bold and shy. And this is the one that I will study. And we know that some personality traits are lateralized. And for example, it has been found that bolder fish are strongly lateralized compared to the shyer fish. And it has been shown in the rainbow fish, but also in guppies. So I think is kept one slide. Okay. But the other part of my experiment is about the parental effect. So we can understand in the words that are affecting the parents, the offspring in an indirect way, so at the phenotypic level. And we know that in a wide range of organisms, if the mother of both parents are exposed to predation, they influence the behavior of the offspring in a positive way. So the anti-predator defense is better in the offspring from parents that were exposed to the predator. But we also know that there are results the other way. So regarding the corticosteroids offspring from mother that were exposed to predator, they were smaller, they were shyer, and they were not good at the predator anti-predator behavior. So my questions were, does the exposure of predation and parents is influencing the offspring personality and also brain laterality? But then also if there is a relationship between laterality and personality. And then my expectation were to find more lateralized fish in the treatment group, but also bolder. So our animal model species was the Gasterosteus saculatus, so the three-spine sticklebacks. And it's widespread in the northern hemisphere, and it's wild in the Netherlands. So we use F2 generation of wild fish. And we can see in the image that they show the colors of the sex just when they're in the breeding period. Otherwise, they look the same. And so one research that has been done regarding the paternal programming suggests the father was exposed to predation. So fish offspring from parents from father that was exposed to predation, they were smaller, less active, in really poor condition. And then the only paper regarding laterality in the sticklebacks is regarding them during their breeding period. So they found that fish in the reproductive state were more lateralized compared to the fish that were not in the reproductive state. And also the males were more strongly lateralized compared to females. Other than this, we don't have papers regarding the laterality in sticklebacks. So the experiment when we started was done on the semi-natural ponds that we have outside, and we divided ponds from the control and ponds from the experiment. And then we have done the experiment last year from April to July, so it is when they are breeding actually. So we use other cues of a real predator, so real birds that we had in another tank, and then we use cues of them specifics, and then we chase them with a fake predator 4 times per week from April to July. During this period, we were removing the eggs from the ponds, so then we actually know there is just parental expression and not that treatment was done on the egg phase or when they were offspring or larva phase. So we were moving the eggs from the big ponds to smaller ponds, and then I moved the offspring to the lab condition. So both control and treatment, they were reared in a standard lab condition, the same condition, both groups. So then I tagged them with a visible implant elastomers. It's just a tiny color, like a tattoo on their skin. So then I could recognize them, and then I could really test them again for 2 times. And then I started the experiment from October to December when they were around 4 months old. And I did the laterality test and personality test. But before this, I was interested in about the behavior of the parents because they actually received the treatment. So I was looking if the predation pressure was effective at the parents' level. So then I did the predator inspection test with the same fake birds that I used to chase them during the treatment period. And then I put a safe zone, so with a shelter, and then a focal fish. And then I record the test for 10 minutes, and then I score how much time they spent in the predator zone and safe zone. And here we have the results. So the first graph is about the first trial, and the second graph is the second trial. And I calculate the mean time spent in the safe zone, so in shelter. And we can see that the treated fish, so the red bar, it's so they spend more time in the safe zone. We'll say that it's, I mean, this is what we're expecting. So then we can say that the treatment increases the shyness. And even in the second trial, the results is the same. So at this point, before starting the experiment at the offspring level, I measured them when they were 4 months old, and they were in the same standard condition in the lab. And then I measured the standard length and the body mass. And we can see that either the standard length and the body mass, the treated fish are way smaller than the control fish. So in this case, we have an effect, an indirect effect of the treatment that we have done at the parents level. So then we go to the offspring. So I did the personality test, the predator inspection test. It's similar to the one that I did to the parents, but in this case, I used the real perch. And then I attached another tank with the focal fish, and then I scored the time spent inside the shelter and then outside so that they could see the predator. And the results, so again, the shyness, so the time spent in the safe zone. And we had the first trial and second trial. In the first trial, we can see that the treated fish, they behave the opposite of the parents. So they say less time in the shelter zone. And the difference between control and treatment is significantly different. While in the second trial, we can see the difference is still there, but it's not significant. So the treatment in this case decreases the shyness, while we see that for the parents, it was the opposite, so increased shyness. But then I look at the correlation between the first trial and the second trial because the fish were tagged, so I could recognize them between the first and the second trial. And it is significant just the treated fish. So they are repeatable over their behavior, while the control, they're not. And then I did the laterality test. So it's a rotational mirror test. So there is like a mirror foil attached to the inside part. And then I score for 10 minutes which eye they used to look at the image. So either left, right, or both eyes and not lateralized, or they were just going around and looking at the mirror. And then I scored the laterality index and then the absolute laterality. So it means just either lateralized or not. And here I use the absolute laterality, but then I also categorize the lateralization. So I use that from 0.75, they were lateralized. Then I categorize them in the graph. And then we had the proportion of population, and then the lateralized or non-lateralized fish. And then in the first trial, there are more lateralized fish in the treated group. While if you look at the second trial, their difference is still there a little bit, but it's not significant anymore. But then if we look again at the correlation between the two trials, only the treated fish are significant. So they correlate between their two trials. While the control, they're highly not significant. So they really change their laterality. Then I tried to do a link between laterality and personality. And then I use in the first graph the mean time of the shyness, and then again the laterality categorized, and there's no link. While if we look at the second graph, is the mean time spent in the predator zone. So we can say boldness. And then again, the laterality in the category way. And it's still not significant, but close to. So I would say that the more they're lateralized, the more they're bold. But it's not significant. So for me, I haven't found a link between personality and laterality. And then just to conclude, I would say that my results are in line with what we can find in the literature. So in this case, regarding personality and laterality, it has been found that fish from high predation zone, they were more lateralized and bolder compared to the fish from low predation zone. But these results are with the fish that they tested, they found in the high predation zone, while my results are from the offspring, so are indirect effect. And regarding the predator exposure, we know that they found that fish from offspring, that the offspring from father that were exposed to predator, they were smaller. And I found the same because my fish were smaller from the parents that were treated. And we also know that predator exposure induces the behavior repeatability. In this case, we found on sticklebacks and personality. And we found the same on laterality and personality. So the fish from the treated group, they have the behavior, they correlate between the first and the second trial of their behavior. And I had some preliminary results regarding the concentration of cortisol. And I have found that there is higher concentration of cortisol in the eggs from treated parents. And the future, it will be that I will analyze the level of cortisol in the plasma of the parents, just to conclude the experiment. Now I'll pass to Paolo.
Can you hear me? Yes. Mute yourself. Okay. So, as a follow-up experiment, I was interested in seeing the effect of the environment on the development of the siblings of the same fish that Flavia tested. And I did this along with a third PhD student, Panizzon. So, yes, to do this, we use this way. To do this, we use the offspring of other offsprings of the same parents treated as Flavia said, and we rear them into some cages, continuing the predation treatment in the same way, so with predation cues, predation odor, and other cues of dead conspecifics. We continue the treatment, it's the same as the parents, but we added also a treatment on size using small and large group sizes. More precisely, the small group sizes were 5 fish and large group sizes were 15 fish. In a full factorial design. And this is why our expectation for this, yes, were that regarding the predation treatment, this would have affected the strength of laterality, as it has been found in previous literature with predation treatment affecting the strength of laterality in a positive way. And we had also expectation for the density treatment, because high density groups, we expected them to influence the propensity of the individuals to take risks due to an increase in competition inside the cages for food. And we also expected it to affect the size of the individuals, because it is well known that fish, when reared in smaller spaces, when they perceive that there is not much space, they tend to grow less. So to do this, we use the same bonds, different bonds, but the same kind of bonds as we did to treat the parents. And we used, as you can see, 6 bonds where we continued the predation treatments and 6 bonds where we continued the control treatment, so no treatment. And in each bond, there were 4 cages, 2 with small group sizes and 2 with large group sizes. And then we wanted to test them both for laterality and for boldness, and we did it this way with the predation inspection test. We used a model predator and some mirrors. So this test lasted for 20 minutes. In the first 5 minutes, we used a removable barrier to keep the fish in the mirror part of the arena, and we checked and we scored the time spent looking with either the right or the left eye, or with no eye preference at all. And then after 5 minutes, we removed the barrier and we scored for 15 minutes the time spent next to the predator model or in the safe area. So we assume that the most far away area was considered safe by the fish. And we repeated this test 2 times at a distance of 3 weeks between the 2 trials. And in total, we analyzed 223 individuals with these numbers between the 4 different treatments. And as results, well, first, the effect of treatment on size, as you can see, there was a negative effect of density, as we expected, but also a positive effect of predation on the size of the individuals. And these 2 effects apparently didn't interact with each other. They were quite independent. Then we checked for the effect of this treatment on laterality, and we found an effect in the expected direction on the first trial with the P plus D minus fish having a stronger laterality. But all these differences in the second trial of the predation inspection test basically disappear, as you can see. Then we checked for the effect of treatment on boldness, on personality, but basically we found no clear effect on personality of our treatments. And finally, we also checked for the correlation between laterality and boldness. And we expected this correlation to be not only with strength, but also with direction of laterality. And we found no correlation with the direction of laterality and a positive correlation with the strength of laterality, but only in the second trial. So, to conclude, density affected size as we expected, probably due to the limited rearing space of the large group sizes. Predation affected also size, both length and weight, in a positive way. And this could be because growing faster could be a way for the fish to escape predation from smaller predators. And the effect of predation on laterality was in the expected direction, but it was interesting that this was seen only in the P plus D minus group. So in the groups where low density, in the group with low density and predation cues. And the relationship between boldness and laterality was a bit surprising because we found no correlation with the direction of laterality. And we found this correlation only in the second trial. Now, I must say that we stopped the predation treatment between the 2 trials of the predation inspection test. So in a way, it's like the predation treatment hid this correlation, and in a very plastic way, when this treatment was stopped, then this correlation apparently was evident in our fish. And the fact that the correlation was only between the strength of laterality and not the direction and personality could be explained because we didn't find any population bias in the direction of laterality at the population level.
Okay, so just to really give another summary of what we found and the difference between us. So my research was based on the parental effect. So I just read the parents and then look at the behavior of the offspring. And then I found regarding the size, the treated fish, well, the offspring from treated fish were smaller in the body mass and then standard length. Regarding laterality, I have found that there are more lateralized fish in the offspring from treated parents. And then personality, I have bolder fish from the treated parents. And then I have found no link between laterality and personality in the offspring. And then regarding the hormones and the preliminary results that I have found more cortisol in the eggs from treated parents. While Paolo, that has environmental effect.
Yeah, well, we used the environmental effects. So we had also, we used fish which parents were also treated, but we used the treatment of the parents as a sort of reinforcement for, as a sort of reinforced environmental cues, at least for the predation treatment. And we found an effect of density, a negative effect of density, and a positive effect of predation on size. We found an effect of predation on the strength of laterality mediated by density. And we found no effect on personality of our environmental treatment. And we found this link between laterality and personality with strength, but only in the second trial when the predation treatment was stopped. And that said, thank you for the attention. And if you have any questions, we are here.
Thank you. Whoa, the hands. Thanks for an excellent talk. I think the mechanism you're talking about, you know, as it goes from parents to the young is cortisol. But I'm working with somebody who's interested in epigenetic changes. So could that be also a mechanism causing changes in body size and brain structure and so on?
I answer. Okay. So yes, it could be an epigenetics modification. But then the thing is that we haven't really treated the eggs and the offspring. So it's just really about, I would say about hormones, because, you know, the sticklebacks, they have maternal and paternal care. So the mother is laying the eggs, but then the father is going close to the eggs, is protecting the nest with the eggs. So probably during this treatment, the eggs were there, and then the father was maybe urinating, and then there was more cortisol in the urine. So that was my hypothesis regarding just the hormones. But of course, yeah, parental effect are also defined as epigenetic modification. So I don't really know how is going the effect. But I would say from hormones, also because I found more cortisol in the eggs. Thanks.
Thank you both for this very interesting talk. So my question is for Flavia. So how do you explain the lack of correlation between your first and second trial in your control group?
Okay, so regarding the link or in general?
Regarding laterality.
Okay, so I would say that the only problem, or I would say the only result that I have regarding the control is that they change continuously their behavior between the first and the second trial. I don't know why. I would say that just because they're explained in some other papers that really the predation is affecting the repeatability of their behavior and actually the personality in some way. So I would say that the treatment in this case of the parents and then move to the offspring, and then yeah, in some way the control fish were just really random. So they were changing a lot. They were sometimes the opposite between the first and the second trial at the individual level, but also the population level, because I checked for both. While the treated fish at the population and the individual level, they are more consistent. It could be maybe also about the hormones, because they have more cortisol in the treated eggs. So it could be also related to the hormonal part. But yeah, and I cannot really discriminate if it was mother or father care during the treatment. So I just say parental effect, but maybe there's also a difference between the mother and the father in the treatment, and then the care of the eggs, and then the results of the behavior. Thanks.
Thanks for the interesting talk. when you look at the Flavia effect and the Paolo effect, I was actually wondering whether, the missing link between personality and laterality is because you're actually measuring or correlating traits factors with state factors, whereas in the environmental, so in Paolo, it will maybe explain because there you measure state and state more. You know what I mean? So I think we all came for mental, I think for buying effect, is always, when we're talking about the trait effect, but I don't think that's actually true. But obviously the personality measurement are certainly trait. So except maybe the explanation why we sometimes find correlation and sometimes don't.
Thank you. You're suggesting that because I did basically the same kind of test, and I basically checked for correlation between two traits inside the same test, so that the fish had a similar state, very similar state, similar emotional state.
Especially when we can find a link between your personality measure and naturality, maybe you're comparing states and or you're correlating traits and states with each other, and they're obviously less likely to correlate than when you have two state measures that are related. So,
Okay. No, it's
Oh, you may have to ponder a little bit on it and then discuss it with Marcus afterwards. I'm very sorry, but we can't take the questions that we have here. You need to rest it out with.
I have a poster anyway also.
There you go. There you go. Come to the poster. Okay, next up is Tim. And he's going to talk about dogs. Well, thank you very much, Mark.